Hey friends, so today we are going to solve some biology questions that I have taken from past papers like from 2020 to 2022. So I'm going to show you how you can solve them in the first place. We are going to talk only concept. If one option is wrong, why it is wrong. If this option is correct, why this is correct. So just let's go to screen and solve them. So first of all, this is the cover page. So now the first question, that is, although it shows two, but this is the first question. Which row shows three structures found in healthy human male level cell in order to size from largest to smallest? So first of all, you need to know that what examiner is asking. That's the tricky thing. This is the 2020 question that appeared in IMAT 2020 paper. So you have got these 5 rows. Just this is the tricky thing because there are like 5 options. You have to choose one. So now just see which row shows 3 structures found in healthy human male level cell in order of size from largest to smallest. Let's first of all understand what the examiner is asking. So he is saying it's human male level cell, it's male, like he has got if it's male, then like there is option in Y chromosome, X chromosome. So you have to assume that in the male there is X and Y chromosome both appear. So no need to confuse about either X will be present or Y will be present. So both will be present in human male level cell because male, he has got Y chromosome from father and X chromosome from mother. So now I just neglect that. If assume that there are no mutations in the cell, there is no it doesn't make any sense. But now we have to identify, we have to arrange that first largest structure appears, then the smallest, then smallest. Okay, we have to arrange in this way. Now let's see. So if we go to first row, then Y chromosome, and then nucleosome, and then histone. Yeah, first of all, you need to know what is histone, what is nucleosome, and chromosome, obviously. So histone is actually a protein. DNA wraps around histone and it makes a thread that is called as nucleosome. When that nucleosome wraps around itself and makes so many turns, then it is called as chromosome. I have got a picture. I will show you in a moment. So now first row, Y chromosome, it's chromosome. Yes, chromosome is in bigger size, and then nucleosome, that's the nucleosome is in smaller size. Then histone is the, I said you, histone and DNA will combine and form nucleosome. So histone is also like smaller than nucleosome. So this is this option is true actually. So the second option, that is nucleosome like histone plus DNA, like a little bit smaller size. Then it has got X chromosome. X chromosome is large actually, and nucleosome is actually, let's say, is smaller than large. So this abrupt or this doesn't make sense to our question, because they said that you have to you have to classify from largest to smaller size. But here the smaller size first, and then larger size, and then Y chromosome. And if you don't know about X and Y chromosome, X is a big chromosome and Y is as compared to small chromosome as compared to. So this is actually also small. So this option is wrong. This third option, histone, small, and this is the smallest. Let me put M with it, smallest, and then nucleosome is small and X chromosome is large. So this is from smaller to larger it is going. So this is also false. And then we have got the fourth option that says Y chromosome is small, X chromosome is large, and then histone small. So this is also reverse our order. So 5, X chromosome large, and then smallest, and then small, like large, small, and then large. So this doesn't make our sense to our question. So this is also wrong. So only one option is true. So your option is supposed to be A. Now you have got what examiner asking. You have to first understand that. Now let's move on to answer. So this is the answer. A was the option. A was the answer. And now here I have got to show you. So this is the DNA. Now when this is in yellow color, this is the histone. When DNA wraps around it, this will be nucleosome. and we have got like HP1, HP2, sorry, what I'm saying, H1, H2, the linker, histone, all that. There are like 5 types. I think yes, they form like 4 in the formation of nucleosome, and these one appears to interlink the linker. It is called linker. You must study that DNA replication if you haven't studied yet. Okay. Now a nucleosome and this DNA is the this has made it nucleosome. Now this will get like more turns and more turns than chromatin fiber, and then it will form ultimately chromosome. So this was the actually order. So if I forgot to tell you who I am, I'm Sami. I've been in IMAT 2023 and got 46.2 marks. So now I'm making videos on IMAT, if you didn't know in case. Now the second question is, which of the following statements about healthy living organism are correct? So this is a very pretty basic question. I have like arranged with basic and medium and then a little bit harder questions, so you must understand this. Now we have got options like A, B, C, D, E, 5 options, and now first you have to choose these questions tend to be a little bit trickier, but this question is very to the point, or let's say easy. So which of the following statements about healthy living organism are correct? First statement is, they are made up of one cell. Yes, healthy human beings are made up of one cell. This is true. You must study cell theory. When you study cell theory, you will understand that this is the basic point. They are unicellular or multicellular. Living organisms are multicellular or unicellular. They have DNA as a genetic material. Yes, this is also true. DNA is found in the nucleus. Then we have got the third option that says they use energy to stay alive when their surroundings change. Yes, this is the homeostasis. This is also chapter included in this in IMAT syllabus. If you don't know about IMAT syllabus, I have like made it very easier in a 1-hour class something. You can watch that. But now they use energy to stay alive when their surroundings change. Yes, absolutely. Now 4, they undergo phases of growth. Yes, they a living organism, you see the baby borns and it goes bigger and bigger and it becomes adult, then older person, then dies. So there are phases of growth. So this is option these options are all true. Now what we have got, option B is the truest. So there was no trick at all. You have to understand what examiner asking, and like this is biology, and biology requires lots of knowledge. If you don't have knowledge, how you are going to do that? Now if you don't know about this was pretty basic, you would have studied high school. If you don't know about these things, then you might not be attempting IMAT in the first place. But I'm not here to criticize anybody. I'm just here to motivate you to study and help you all that stuff. Okay, now they undergo phases of growth, everything. This was like easy question. So now move on to, so the option was all statements were true. Now the third question, that is, which of the following could be found within a molecule that has just been made by transcription in a healthy human pancreatic cell? Literally, you don't need to know about pancreatic cell, because here it doesn't make any sense, because all cells, it's just generally asking about cell. But no need, you must know what is pancreas, and it is made up of like acinar cells, and then there are isolates of Langerhans, alpha beta cells. Alpha produces glucagon, and beta cells produce insulin. You must know that, because if you don't know them, how you are going to sit an IMAT exam? But which of the following could be found? So we have to underline. So which could be found within a molecule that has just been made by transcription in a healthy human cell? So first of all, the trickiest thing is you know what is transcription. If you don't know what is transcription, then you can't answer this question really. Okay, what is transcription? Transcription is when DNA, this is a central dogma of life. If you haven't studied, you must study that. That is very important. DNA converts into messenger RNA then goes into cytoplasm. This happens in nucleus. Transcription happens in nucleus, while translation happens in cytoplasm. Messenger RNA forms and it will go into cytoplasm, and then binds with ribosome. Transfer RNA will come, and the information will be read, and amino acid protein chain will form. Now transcription is first of all DNA. The information on DNA is like stamped, like on what to say, messenger RNA. The DNA information is transferred to messenger RNA, not transferred to, actually the messenger RNA forms by reading the information from DNA. Okay, now what happened, that is called, the DNA strand will be called as a template strand. Okay, now we have to know about, so now here for confusion they have given that now it means that the molecule that has formed it is RNA. Okay, RNA has been formed. I am just like explain. I love explaining, literally I love teaching actually. So now first of all RNA form, so you must know what will be included at RNA. If you don't know about RNA, then that's the basic thing. You must literally know about, study deeply RNA, types of RNA, ribosomal RNA, transfer RNA, and blah blah blah. Adenine. So adenine will be present. The only thing that absence in RNA, that is thiamine. Okay, thiamine is not present in RNA. Would in case of thiamine we have got uracil. Okay, that's the like tricky thing. That's why they confuse with you adenine. So when you will be in exam with pressure, you will like forget about adenine. Okay, secondly, peptide bonds. Actually, it's not a protein, that's DNA. There will be hydrogen bonds, and it's not DNA actually, it's RNA. It will not have peptide bonds. Instead, it will have hydrogen bonds and phosphodiester bonds. So phosphodiester bonds will be present, I've just told that. And the ribose sugar, ribose is present in RNA, while deoxyribose is present in DNA. So 1, 3 and 4 only, 1, 3 and 4 only. So D option will be true. Have you got? I just kept explaining. So this is my Instagram handle. If you want to follow me, go just follow me. There will be like cool stuff. Okay, now this was the thing, like D was true. So I just want to show what is phosphodiester bond. So this is phosphate that forms OH bonds with the OH on carbon number 3, and here with this carbon with the 5, so it makes phosphodiester bonds, like one ester bond this and this, then diester bond with phosphodiester bond. You know that actually these bonds will at least in all exams, the format, there is like one question from DNA and RNA, like which bonds are present, which nitrogenous base is absent, everything. So you must know that. Actually, you must learn that. The diagrams each show a section of the cell surface membrane to illustrate the fluid mosaic model. So if you have seen the picture of cell membrane, then you can easily answer this question in a like moment. But if you haven't, then this gets a little bit trickier. But don't worry, first of all, you don't have to panic. Just understand what it is asking, and then try to retrieve your information from whatever you have studied. You must have studied. Don't, like in real-time exam, you will forget everything. So now you have to identify which is the correct model of fluid mosaic model. If fluid mosaic model was the most acceptable model of cell membrane, I think that it was presented in 1972 or 1957. I don't remember. It's 1 year. But anyways, fluid mosaic model. Now fluid mosaic model, let me explain you if you don't know. In case, fluid mosaic model is lipid bilayer. Proteins are embedded. Some are like going out and inside. It is intrinsic, and some are like floating or on lipid bilayer, that are called extrinsic protein. Yep. And then now on lipids, in a lipid bilayer, the carbohydrates that will be attached will form what will form actually? What will form? Glycolipids. You are like just like me, glycolipids, and with protein they will form glycoprotein. So here you go, this is glycolipid, and the protein, this is the protein, so this will be glycoprotein. Yeah. Actually, now here they have got a key that they gray or like everything you need to know. Gray shape represent protein. These are proteins, and these black bars are they called as cholesterol. Now here's the tricky point. I'm going to make a question. Phospholipids give dash to cell membrane, while cholesterol gives dash to cell membrane. Just type in the comment. So now here you go. This is the cell surface, so it will be like outside of cell, and then here is the cytoplasm. So it will be like cell surface here, and then cytoplasm inside. So it's the cell cytoplasm inside, and then here goes cell surface. So now you have to remember that glycoprotein, because first just picture, if you don't remember, because fluid mosaic model is very important in when you will be studying cell biology. When you will be studying cell biology, that is very important. So now understand it. You have to remember that the phospholipids, this is the phospholipids, these phospholipids head will be on cell surface, and then inside the head will be inside cytoplasm, because head are hydrophilic while tails are hydrophobic, so they will arrange in this way. Now what will happen, this is the one thing, like head will be like both tails will be facing each other, okay? Both tails will be facing each other, but it can happen like head and head are facing each other. It can happen. You will see that there is the where you have to find that heads are facing tails. Actually, where is it? So here you go. Heads are facing heads. This is like inappropriate. Just straightforwardly cancel it. So now, first of all, let's see which one is correct. Actually, so we will come to correct later. Now, what is A? So A has got like great, and this is cholesterol. Cholesterol will be present like this with phospholipids, and here with the glycolipids will be outside of the cell. Always remember, on cell surface glycolipids and glycoproteins will be present. Okay, so this seems kind of true, like looks good. Now cell surface, here you have got what you have got, just straightforwardly the glycolipids, or what to call glycoprotein, lie inside, so this goes wrong. And then in C option we have got cholesterol. Cholesterol can't be like goes from inside to outside. That's like, don't see any other thing. Just if cholesterol is going out and in, that doesn't make sense. Just straightforward cross it. Although you will find something else in this, but this is the this can't happen. In D option we have got what we have got, again inside the what we said earlier, the glycolipids, glycoprotein, and then here glycolipids can't be. Glycolipids and glycoproteins are cell surface markers. You see antigens or RBC, these are cell surface markers. These will be lie outside, so that this can't happen. So this will be cross, and so option A is true. Really, option A is true. Wow, option A is true. Now we have got the another question that says the SRY gene is normally located in the home and Y chromosome. So SRY gene is present on Y chromosome. Y chromosome is in male, so that is responsible for male characteristics. Like male has got beard, yeah, and then male has got shrill, grave sound, and then male has got hair on body. So these are the appearance of SRY gene, and then we have got genitalia, that is due to SRY gene, that is for maleness. It makes maleness. Now SRY gene is normally located in human Y chromosome, so that is you must know that. Actually, this gene initiates the development of male characteristics. So gene initiates the development of male characteristics, primary and secondary sexual characteristics. Using this information, which of the following statements about the SRY gene is correct? Okay, now see, first statement is, so you have to find the correct one. This is the important. Just always encircle whatever you are going to answer. Insertion of the SRY gene into an X chromosome of a female zygote could give it male characteristics. Yes, absolutely. If SRY gene is on X chromosome, SRY gene is responsible for maleness. So if it's on female, it will definitely give male characteristics. So this happens to be true. Now second statement says a sperm cell arising, sperm like from male, arising from incorrect division, which has functional SRY gene. Okay, it has got a functional SRY gene, could give sperm has got with incorrect division, it has got functional SRY gene, could give rise to a female with a sex chromosome trisomy following fertilization with a normal ova. Actually, this is not possible. Now, if I said and give you explanation, that will take like ages, because I could not. I don't have pencil that I could write, but anyways, so the basic thing is that actually I will pin a statement in the comment that you can read to understand it more clearly. Otherwise, I will when changing, you will like get confused. Okay. Now the third statement is during meiosis. So during meiosis, that's the like cell cycle. If you haven't studied, do study it. Division of during meiosis, crossing over could result in the formation of X sperm with the SRY gene. Yes, X sperm, like it is any sperm with the SRY gene, which would cause male characteristic after fertilization. So what happens in during crossover, that happens in prophase 1. So what will happen, that when crossover will occur, exchange of genetic material occur on chromosome, when they chromosomes. What will happen, that if SRY gene from Y chromosome gets on X chromosome, and now X chromosome is responsible for femaleness, actually it will form female. Now when now X chromosome has got SRY gene, when it will go into female, this will cause male characteristics, because SRY gene is responsible for maleness. So now it's on female X chromosome, so it will play its role, like it will secrete testosterone, and it will form like lots of other things, primary and secondary, all those things. So this happens to be true again. So option 1 and 3 are only true. So B option gotta be true. Yeah, B option gotta be true. Now this question has got the occurrence of which of the following could allow you to distinguish between the processes of DNA replication and transcription. So now you have to find, first of all, you must know that what examiner is asking, what question is literally asking from you. The occurrence of which of the following could allow you to, actually you need to differentiate between these two things, DNA replication and then transcription. You have to differentiate. DNA replication is DNA replicates, the form is exact replica. Transcription, a messenger RNA or RNA forms from DNA. Now you must know that there must be a thing that would differentiate DNA from DNA replication from transcription. There could be a thing like what happens in DNA couldn't happen in transcription. I can't remember at the moment, but let's see if we can find in these three statements. The breaking of hydrogen bond. Breaking of hydrogen bond will occur in both DNA replication and RNA replication. DNA replication, DNA will open up and one strand will form here, one strand will form here, and so the other strand, there will form one strand. So two strands open up, one strand will form with this, and second strand will form this. So hydrogen bond break and strand open. So while transcription, DNA will also open up and one will form the other thing. So hydrogen bond break. And then after, when the formation of hydrogen bond will occur, nitrogen's bases form hydrogen bond, so this will occur. Actually, this occurs in both. The breaking of hydrogen occurs in both. The formation of hydrogen bonds occurs in both. The formation of phosphodiester bonds occurs in both. In DNA, it also occurs from 3 to 5, and in RNA, it also occurs 2 to 5. So 5 to 3, 3 to 5, whatever you call it, because actually in DNA we have got 2 strands, in RNA we have got 1 strand. So these occurs in both of them. So it means that there is nothing that can distinguish between DNA replication and transcription. Like there must be a thing that occurs in DNA replication and does not occur in transcription. So we haven't found anything yet. So here we gotta go. None of them is like, we haven't found anything. So none of them distinguishes between DNA replication and transcription. So this was like pretty easy. It wasn't hard as I recall, as I say. Okay, a single base mutation occurs in a gene. So if you don't know mutation is a very lengthy, I would say, one chapter that you have to cover up, because there could be one question that is important. Actually, there is like trisomy. There will be actually point mutation and then substitution, and there will be like deletion, inversion, insertion, and all those things. You have to like study them and remember them, because these are the things that you have to remember. So a single base mutation occurs in gene. So mutation has occurred in gene. So single base mutation is also called point mutation. Okay, point mutation is also called as what I'm writing, whatever it is I'm writing with mouse. So point mutation in a gene. Which of the following are possible effects on the protein? Like gene will produce protein. How I have explained you, I can like explain you, it's not going to be lecture, but I'm thinking about those things. Like I should deliver some sort of lectures that you will know shortly after this video, or maybe just upcoming video. I will announce what I have got in mind. That will be absolutely mind-blowing. So stick with me, just follow me on Instagram, you will know that. Which of the following is possible effects on the protein? Like what will be effect on the protein that will be formed from that gene? Now the protein has an altered tertiary structure. Yes, absolutely, the protein has an altered tertiary structure. So what will happen? Actually, if mutation has occurred, the protein has an altered tertiary structure. In this way, the primary structure will fold around itself. Alpha helix and beta pleated sheets will form secondary structure, and then it will get in 3D structure, that will be tertiary structure. So if the point mutation happens to be in this way, that it interrupts those folds and forms like linear things, and just those disturb that 3D structure, that will be like tertiary structure will be altered. So that can be true. The protein is shorter in length. So protein can be shorter in length because gene form the actually, if that gene where mutation occur, there happens to be a codon that codes for a stop codon, that forms the amino acid that will like cut. How I should explain? I can like go in very deep. I don't want to go in that because this video is already getting lengthy. So what will happen if there is a stop codon that codes happens to be stop codon, then there will be like no continuation of protein chain, so that will block and protein chain will become shorter. So if before mutation, that would be like 2 cm, and after mutation the stop codon comes like after 1 cm, the protein length has decreased. So this is how it is played. Actually, I can explain more, but I don't want to do that. So okay, the third option is the protein is unchanged. Yes, the protein can be unchanged if that if in that gene, whatever the gene was, so if mutation occur in this way, that it forms the codon that is going to code for the same amino acid, because their amino acids are coded by multiple codons. So if that codes for same amino acid, then the protein can be unchanged. So all these options are true actually. So the B is going to be the best option. You have to choose the best option actually. Okay, now this question, I don't know which is this question. The heart of developing human fetus, when it's the from 4 to what week? Actually, that's the what is the fetal period? What is the fetal period? Just get it. I know that, but I just forget at the moment. It's from fourth week embryonic period. Actually, yeah, 4th week to 9th month. 9 months. No, from what was that? Actually, I forgot that. Embryonic period, and then there is fetal period. There is a specific actually time period from which week is to which week. You should search about it. I am not responsible for searching everything. No, I'm just kidding. The heart of developing human fetus actually normally has a hole. Like now baby isn't born. Yeah, let me clear you in easy words that fetus is something baby will born after 9 months. So before 9 months, when it is going to born, that is fetal period. Now fetus normally has a hole in the septum. So in the septum. The septum separates the right atrium and left atrium. So septum is going to be a separation that is going to separate one or the second thing. So in this case here it is separating right atrium and left atrium, right? So it's the heart. So like this is the heart. So now above are the atrias, right atria, left atria, and then below are the ventricles. So if you can see my fingers actually in camera, yeah, above are the atria and then below are the ventricles. So now what happens is that here is going to be sort of septum. So now what happened, which one of the statements about the circulation of the developing fetus will be a hole in the septum is correct? So here there gotta be septum, but here is the like sort of a hole. Now blood can go from right atrium to left atrium. So what happens now, if I like give you a brief of circulation, the blood will come, the deoxygenated blood from body to right atrium, then it will go into right ventricle, and then it will pump to lungs where it will oxygenate, then it will come to left atrium, and then from here then left ventricle, and here it will pump out to the body, whole body via aorta. So now what happened, if you know now these things, blood entering the right atrium can bypass the lungs and go to the rest of the body. Blood entering the right atrium, yeah. Now deoxygenated blood is pouring into right atrium. What happens, there is a hole in between right atrium and left atrium. Now it means that blood can flow from right atrium into left atria. It means that deoxygenated blood will go into left atria, and then here it will go into left ventricle and will spread into whole body. So this is actually true. It will bypass lung. This is true. Blood in the right atrium may return directly to vena cava. So that can happen. How it can happen? This isn't, because there is a not hole in vena cava and between right atrium. There is a hole between. Although there is a hole, blood will come into, but there are like walls. That can happen. This isn't true. The third, the C is the AV node, the atrioventricular node, will not cause ventricle systole. Why? There is a problem in AV node. We have got any problem. There will be like conduction from SC node to AV node, and then it will spread from bundle of phase to Purkinje fiber, and then Purkinje fibers, and then systole occur. So that has not got any problem. The atria, the conduction system will work as fine as it was. There is a hole in between atria and ventricle, although and in atria and ventricle there is no AV node is not present. It is like on the apex of ventricle the AV node is present. There is an annular ring around ventricles and atria. You might know that why I know all these information. In our med school there is like heart, and we have studied very in depth, so I know that. Also I know at my time of IMAT I had studied very in depth heart, because I love literally heart. I love heart. I love heart, just the circulation and conduction. Okay, although I love heart too in other sense. Okay, this isn't true. Blood in the pulmonary artery will go directly to the aorta. So no, pulmonary artery will take blood from right atrium to lungs. So it can go into aorta. There is not like hole in aorta and pulmonary artery. So that is not true either. So the maximum pressure in the aorta is the same as the maximum pressure in the contracting atrium. It's not true actually. I don't want to go into debate about pressure, because that will confuse you. Now just can give you the, if you know about valves, the valves open due to pressure changes. So if pressure in atria will like rise, the AV valve, if it's right atria, tricuspid valve will open and blood will pour into right ventricle. So when the pressure in right ventricle will get very high, the semilunar valve in pulmonary artery will open and blood will pump to lungs. So now here how it happens. So the maximum pressure in the aorta is the same as the maximum pressure in the contracting atrium. So this is true, because in atria just can give you like a very hint that you should remember for the rest of your life, that pressure in aorta is very less as compared to aorta or ventricle. Just remember that there is like a very huge difference. In aorta the resting pressure is 80 what is mercury atm, actually atm. No. 760 80 1 ATM is equal to 760 HHMMG. What is it? I don't remember, but whatever it is. So HHMMG. So there is a like huge difference. So in resting, aorta has got 80, and in during systole, or when blood is coming from left ventricle to aorta, there will be 120 mmHg. Sorry. I just forget all that. Due to tension, I'm recording whole video, so that's why I got tension. mmHg. There is resting. So in atrium, there is like 2 to 3 mmHg in right atria, while there is like 6 to 7 mmHg in left atria. But when it comes to ventricle, there will be 120 mmHg. That's your blood pressure. Systolic blood pressure is what? It is 120 mmHg. So now, and diastolic pressure is 80. You know that. If you don't, I know you don't know, but whatever it is, there is not going to be pressure, same pressure. So the only option A is true. So the second thing, option A is true. So now we have got the last question. I think, which one of the following, we have got one more question. Which one of the following statements is correct about the cardiac cycle of a healthy human? When the left ventricle relaxes, the aortic valve closes. So first of all, let's see what actually will happen when the left ventricle relaxes, the aortic valve closes. Now, when the left ventricle relaxes, when the left ventricle relaxes, when the left ventricle, now ventricle is relaxed, what will happen? Aortic valve will close. Yeah, that's the statement. Now, if the pressure when the ventricles are contracting, ventricles contract, now blood have to pump, so aortic valves will open and blood will go out. So this is true actually. So B option is when the right ventricle contracts, the right atrioventricular valves open. When the right ventricle contracts, the right atrioventricular valve opens. When the right ventricle contracts, the right atrio-AV valve opens. Actually, yes, it's happens to be true, but why it's right? No, actually, I was thinking about atrium. When the right ventricle contracts, will contract, the AV valve have to be closed to stop the backflow of blood. Because if valve didn't close, for example, in left ventricle there is blood, now it has to pump. When it will pump, it will go into pulmonary artery. But if to go into pulmonary artery, there must be the above valve, the tricuspid valve, AV valve that is called, must be closed. If it didn't close, then blood will go into aorta and less blood will go into, then there is like, will get clinical, that will be pathology. But now, so blood, the AV valve has to be closed. When the left atrium contracts, the aortic valve opens. When the left ventricle, the left atrium contracts, the aortic valve opens. No, it's not a sense, because only when left atrium contracts, the AV valve, not aortic valve. When left ventricle will contract, the aortic valve will open. So atrium doesn't make sense. Then D, when the right atrioventricular valve closes, the left atrioventricular valve opens. No, both opens at the same time and closes at the same time. When the right atrium contracts, the left ventricle contracts. When the right atrium contracts, the left ventricle contracts. No, atrium contracts at the same time and ventricle contracts at the same time. So that doesn't make sense either. So only option A is correct. So option A is correct. So now we have got the last question, that is, which of the following is a feature of HIV? That is, acellular must study about viruses, study about the cycles of HIV. That's very interesting and very good. Actually, I have made a notes. If you don't know about my notes, you can go to samicomment.com/IMAT notes. IMAT, the space, whatever the space bar, actually dash, we call it dash notes. samicomment.com/IMAT dash notes. So it has captured which are the following features of HIV. It has capsid. Yes, proteinous structure around it. This has got a capsid. Virus got capsid. HIV specifically. If you I remember, because I studied the cycle of HIV, and that was very important, very interestingly. HIV comes and attaches on CD4, then pushes its core, whatever it consists of, its RNA. Actually, this makes this statement false, because it has got RNA, not HIV is a RNA virus, not a DNA virus. Okay, so HIV will pour its core into the T cells. Actually, it attacks on T cell, helper T cells. Now here they will go, and reverse transcription will occur. DNA, the RNA has got, sorry, HIV has got RNA. It will reverse transcript into DNA, and then it will form all the virus thing that it has to survive to make more new copies. So 3, it has ribosomes. No, it don't have ribosome. If it would have ribosomes, it would be far more dangerous, because it utilizes, HIV utilizes the ribosomes of host helper T cells. It actually attacks. So it is going to use the ribosomes of host, not his own. So it has captured. So only option 1 is true. I hope it had made sense. So this was all. I have got cooked, so hope I would have delivered something, and I hope you have learned something. So if you want more, then drop a comment, and also if I was wrong somewhere, then do correct me by writing, or you can follow me on Instagram. So isamikhamar.com, sorry, isamikhamar, samikhamar.com is my site, where you can download all the necessary sources. So I am going to make something very unique, so stay tuned, and just bye till that. So see you later. Now if you want to check this, my playlist of IMAT syllabus explanation masterclasses I have made, so you can check that, and see you in next video. Till bye bye.